Hey Liz, I have a question. What are you seeing right now? Are you just seeing the full screen of my PowerPoint? Did you say something? Yeah, can you hear me, Liz? I can't hear you. Okay, can everyone hear me? Seems like we have some issues with my sound. You can hear me? Okay.
I think it was issues with my sound.
Okay. Fine. Um, so we're gonna continue talking about molecular dynamics. And so let me go ahead and share my screen with you all. Okay, so just to recap, right? So we introduced molecular mechanics as the essentially the energy function that's used to describe the energies in molecular systems, to model molecular systems. And then the molecular mechanics is used to do MD simulations. And also something we're not going to talk a lot about, but also something called Monte Carlo simulation and Brownian dynamics. So today the plan is to continue talking about MD simulations, and I'll actually send you a link to web interface that allows you to run simulation directly from your web browsers, and so then you'll be able to get a feel for what one of these simulations look like, and then also you'll be able to visualize the output. So what I could do as well is probably just give you a view of what an MD simulation produces, right? And so you're all seeing my Primal window? Yep. So what I've loaded here is are two trajectories. And trajectories just refer to the molecular objects that we're going to be simulating as a function of time. So at every time point, we have a conformation, and so we're looking at a set of conformations. And that collection of conformations, when viewed one after the other, we refer to as a trajectory. And so you're seeing two for isopentane and pentane that I generated just a while ago using that web server that we'll use together. And what it produces is this. You can animate this and actually look at your system moving as a function of time. So this is essentially what molecular dynamics is, right? It's a tool that allows you to look at the dynamics of molecules, hence the name molecular dynamics. And so these are relatively simple systems. However, you can apply the same kind of techniques to biomolecules, which are much more complex and much larger. So what I'll show you next is an example of another trajectory for a more complex system. This is actually this is an RNA that was actually talked about in one of the journal club presentations. This is the RNA from the Zika virus, right? And we've been studying this in the context of a project. And one of the things we were doing is applying a force to the 5' end of the RNA, right? So if you turn this around, let's see, where is it? There's this 5' region here. We're applying a force and we're trying to simulate the dynamics of the system. And so what I'll show you is an example of a trajectory that we generated. And so you can see we're pulling on the end, and at some point you'll see that the system starts to deform significantly, coming close to the end of the trajectory. So molecular dynamics is a tool that allows you to get really detailed information about molecular processes, right? When you do experiments in the lab, very often you lose a lot of details, right? And so, and these simulations allow you to try to model in some of those details, because you can imagine from this kind of trajectory, you can ask specific questions about when certain contacts are lost, right? And in what order they're lost. And so this is really critical information when you're trying to build models to explain what is happening in a test tube or in a cell. So this is just meant to give you a feel for, what a molecular dynamic simulation produces, right? It's simply information about the position of all of the atoms in the system as a function of time. So in this particular example, if you look on the bottom here, I have 1,001 frames, and each of those frames correspond to each of
I
those.
was going to ask that question in a second.
Okay, each of those frames correspond to a snapshot in your trajectory, right? So essentially, you start from some starting point. You start from an initial structure, and then you propagate dynamics to the next state, right? And then from that state, you propagate dynamics again, and you keep doing this iteratively. And as you go from one to the next, you get the buildup of this kind of movie.
So Aaron, there's a clarification question in the chat from Dylan. So just applying forces to the 5' end causes these global dynamics?
Oh no, so this is yeah. So there are two sets of forces that are being applied. This is I should have mentioned that. So we have the the forces that are due to the interactions between the system without any external force, right? And you'll still see movement, but in this particular simulation we're running, we've added an additional force, which is a force on the 5' end, and that's the force that allows us to pull the 5' end of this RNA out of this ring, right? So independent of that external force, you would still see movement, but it wouldn't see this pulling of the RNA strand. So thanks for asking that question. So in the other window that I showed you with these small molecules, the forces that are propagating dynamics here are just strictly the forces due to the interactions between the atoms in a system. So let's just look at one at a time. So again, we start from some initial set of coordinates. Then based on those coordinates and the energy function we have, we can, and some initial velocities, we can propagate dynamics to the next state. And then from that state, propagate dynamics again. And this propagating of a dynamic simply means calculating the forces, which tells you the acceleration. And for every atom, you can then propagate it further in a one time step further along that acceleration vector and keep doing that. And the result is these kinds of trajectories. So I just thought it would be useful for you to, get a visual sense of what is produced from these kinds of simulations before as we continue to talk a little bit about the details of the method. So we introduced this before in the last class, which is the force field. And again, this is really critical. This is what determines the forces, which are then used to essentially set up Newton's equations and motions and integrate those equations, right? So different force fields have different parameters, and so they will result in different trajectories starting from the same structure. But the key is to know that what you do is that you typically try to model interaction in terms of you account for some bonded interactions, right? So the bonds are shown here, the angles, the dihedrals, and then you have the non-bonded terms here as well, that accounts for Lennard-Jones interaction and charge attraction and repulsion through Coulombic potential here. So which are all of which we talked about last time. So again, these are just graphical representations of the individual terms. This is for the interaction between two atoms, right, that are not bonded. These are the bonded interactions, and this is the angular interaction. You have energy changes as a result of rotation about a central bond. In this case, you have 4 atoms that are connected, A, B, C, and D, and the rotation is about the central bond between B and C. And this function gives you how the energy changes as you rotate about this bond. And then you have Lennard-Jones, not Lennard-Jones, electrostatics, and then the dipole-dipole interactions. So one of the use of the molecular mechanics of force field or force field in general is that you can use it to minimize or optimize a system, right? So when you determine the structure of a biomolecule using extracrystallography, very often some of the structures, they have steric clashes. And so you actually want to relieve some of those clashes. And so molecular mechanics can be used to do that. And the way you do it is kind of depicted here, right? Imagine your starting structure is this black dot, right? So what you're seeing here is the potential energy as a function of some reaction coordinate R. And R is just, let's just think of R as some molecular trajectory. So this describes the state of your system. So as you change the structure, R would change. Of course, this is a one-dimensional description, which is simplified, but it's meant to kind of highlight how energy minimization works. So what you would do is that you start at this point, and using your molecular mechanics force field, you can actually compute the force. And so if the force is pointing in this direction down, then you move your coordinates in that direction by a small bit. And then so that means you'll update your coordinates to this new position. And then you keep doing the same thing over and over until you get to a region where the potential is at local minima. Now, how would you know that it's a local minima? You can do things like take the second derivative, and based on the combination of your knowledge of the force and the second derivative of the energy with respect to your structure coordinate, then you could figure out whether or not this is a local minima or maxima. So this is just one use of the molecular mechanics force field. Note that the what I described here, this iterative procedure of starting from a state and following the force, the direction of the force, is not molecular dynamics, right? It's really just an iterative scheme that allows you to update the coordinates such that you are able to access a local minima on your potential energy surface. Molecular dynamics requires you to actually propagate dynamics based on Newton's equations, and that's what we're going to talk about in a bit. Now, one of the problems with using molecular mechanics to do energy minimization can be seen in this particular image here, right? So if we started at this location and we were simply following the direction where the force is going to be decreasing, that means based on the steepness of the potential, you would end up at this local minima here, right? And this has an energy of some energy value, which you get by just simply going along here. But note that there is another minima on the surface that actually has a much lower energy. So this here is a local minima, but this is the global minima. So if you start at this point and you use what I described as really a steepest descent approach, you would get to the wrong local minima if indeed what you were trying to do is go to the global minima. So this kind of highlights some of the challenges you face when trying to do minimization, where if you're on the wrong side of a barrier, you can actually you get trapped in a local minima. So let's see. Okay. So the energy minimization is, again, an iterative procedure that allows you to update the coordinates using your energy function. You need to be able to compute forces, and forces is simply the negative gradient of the potential, and the potential is given by your force field, right? Now, there's another kind of dynamics you can do using your energy function, and this is kind of highlighted here, and it's called Monte Carlo simulations. And why Monte Carlo simulations are interesting is because you don't actually need to calculate forces. All you need to do is calculate the energies, right? And so the way it works is that you start from some initial configuration of your system, and then you attempt to make some change. So you change the position of an atom, for example, right? And then you decide whether or not you're going to accept or reject that move. And that decision is based on this acceptance criterion. And that is essentially what you're doing is calculate the energies before and after the move. And if the energies decrease, then you accept the move. If it increases, you reject most of them, but you accept a small probability of those, right? So why might that be useful? So let's imagine we go back to this problem here, and rather than doing energy minimization, we were doing Monte Carlo simulations, right? And let's imagine what we really want to do is actually get to the global minima. And we started from this point here, F1, where it has the F1 arrow. So this black point is where we started. So imagine we make a move, right? And we accept only the moves that decrease the energy, right? then a small move would that decreases the energy would lead us to this point and to this point and then to this point with the local minima, right? Any moves, once we're at this local minima, any moves we make, right? Any small moves we make, they're going to increase the energy. And so what it means is that we're going to get trapped at the local minima. So if we're here and we make any small moves, we can see we go this way, we're going to increase the energy. If we only accept moves that decrease the energy, we're going to reject that move, right? If we went this way, we would reject that move again. So we'll essentially be stuck here. And so the Monte Carlo acceptance criterion has a small probability that you would accept moves that increase your energy. And that will be useful in scenarios where you're close to the barrier, right? So let's say imagine you're at this point and you make some small move and you get to this point here. If you accept it, then you now have a chance to access this side of the hill and get to this global minima, right? So this acceptance of these moves that increase the energy is actually what allows you to escape a local minima, right? So you can imagine being here at some point, or let's say being here, and there is some probability that you can actually walk up this hill and go down this one and arrive at this global minima. So this kind of Monte Carlo simulation strategy is what was used initially by the Rosetta group that developed the Rosetta protein structure prediction technique. And what you're seeing here on the right hand side is a image of the code that they use, but the key steps is that they have some initial structure. So that's what's shown here. They generate some initial structure. This part is where they set up essentially the force field. So that's kind of analogous to the molecular mechanics energy function. Then they set up the simulation parameters that say, I'm going to do a certain number of steps, and I'm going to use the Monte Carlo method to do that. And inside this function, it contains all of the details, right? But again, it's simply make a change, ask whether or not it increase or decrease your energy, use that information to decide if you're going to accept or reject the move. And what that allows you to do is to start from this kind of extended conformation and arrive at a folded conformation. Now, this kind of strategy is very generic. This Monte Carlo simulation technique is very generic, right? You can use it to solve any optimization problem, really, right? The key is that you need some function to optimize on. And in this case, we're using the molecular mechanics-like energy function to do that. And that allows us to go from an extended conformation to a folded conformation like this. And it works really well for very small systems. So it's an effective optimization strategy when you have a simple enough energy function to work with. So Brownian dynamics is another kind of dynamic approach where what you're looking at is more global motions. And so let me see if I can play this. So you're seeing here is the association of a protein, two proteins, right? And so these kinds of simulations are used to study association rates. So if you look closely, you can see like there is no internal changes in the structure of the protein. It's really the translation and rotation relative to some fixed axis. And so this is a case where you are propagating something that looks like equation of motion, right? But it does not include any kind of internal motion inside of each of the domains, right? So here what you have on the right-hand side is essentially saying this is just mass times acceleration, right? It's mass times the second derivative of position with respect to time, which is acceleration. So it says that that is equal to the force that's in the system, plus there's some drag on the system, right? So that's a friction. And then there's also a random component, this RIT here. And that allows you to then generate these kinds of trajectories that describe how two systems might associate with each other. So simulated annealing is a another optimization strategy that is used in molecular dynamics simulation, along with molecular dynamics simulations, but it's a very generic uh, approach, right? That can be coupled with any kind of sampling technique. And the idea is kind of what we talked about a while ago, is that if you have some potential, this is a potential you're seeing here, the total energy is on the vertical axis, the structure is on the horizontal axis, and as you change the structure, the energy changes. And very often what you want to do is to get to the global minima. Question? Oh, okay. So you start from this point here and you want to get to this point over here. And so one of the things you can do is actually run very, very high temperature simulations. And when you run high temperature simulations, what that means is that you have a lot of kinetic energy in your system. And what it allows you to do is essentially jump over barriers, right? And then after you jump over the barrier, you can lower the temperature. And so similar to annealing, the annealing is really the cooling of the system. So you do this kind of simulations where you start where the temperature in your simulation is really high, so your kinetic energy is high, allows you to escape this barrier, and then you slowly cool the system, and that allows you to arrive at a global minima. Now, this is a stochastic process, so every time you run it, you won't always get to this global minimum. But if you run it many, many times, you can always go back and select the one that allows you to minimize the energy. So again, this is a very generic optimization strategy that's used to solve lots of optimization problems. Here, the optimization problem is trying to minimize the energy, go from this state to a lower energy, which is the state here.
Hey, Aaron, I have a
Yep.
quick question. So is this simulated annealing used mostly for like RNA or DNA simulations, or is it also something that you would use to study proteins?
Yeah, this is used. This is just generic, really generic. So it can be used to study any kind of molecular system, and it's also used to optimize problems in when fitting equations, right? So sometimes you're working where you have a nonlinear equation that you're trying to fit. You can use simulated annealing to fit that equation as well. So it's really simulated annealing is an optimization strategy. We're just talking about it here in the context of running simulations to optimize the energy, the total energy, but
Okay.
it's pretty generic. So we could skip this one, right, and talk a little bit about why people care about molecular dynamics, and well, the fact that people do care about molecular dynamics. And so in 2013, the Nobel Prize in Chemistry was awarded to these three gentlemen, and they were all closely involved in the development of molecular dynamics approaches to study biomolecules in general. And so Martin Karplus is actually the postdoc advisor of Charlie Brooks, who is the director of biophysics. So he visited, when was it? I think he visited university several years ago to give a talk, which is quite interesting. So, and let's talk a little bit about simulating biomolecules, right? And what's needed. So what I'm showing you here is in green is your rendering of your biomolecule. In this case, it's a protein. And to actually simulate this in a way that's physically meaningful, we very often try to also include water molecules, right? Because in reality, biomolecules are surrounded by water molecules. So in addition to having models that describe the biomolecule, in this case a protein, you also include models for the water molecules that surround the protein. Now, the way the energy functions work, right? What you actually have to do is for every atom, every pair of atoms in your system, you have to calculate some interaction, right? So you can see there are thousands of water molecules around this protein. and there are probably on the order of 1,000 atoms in the protein itself, right? So if you have to do pairwise interactions, calculate all pairwise interactions, that means that you have to execute many computations, right, or calculations, just to get a single set of forces, right? And so that's why these kinds of simulations are computationally expensive, because it requires thousands and thousands of calculations just to get a single set of forces, which you then use to propagate the dynamics by a small time step, right? And just to give you some context, the time step that's used in simulation is on the order of femtoseconds, right? So that's 10 to the negative 12 seconds, right? However, the time scale of biological processes can extend to the second regime, right? So that means if you want to simulate something, a very long process, you have to actually execute billions of calculations, sets of calculations, right? Because you have billions of time steps that you need to actually simulate. So that's the reason why these calculations are really expensive. Yeah, it is because you have to use very short time steps, and you have lots of interactions to consider. Now, it could actually be a more difficult problem, right? Because what we actually do when we do simulations very often is that we treat a single protein surrounded by a water molecule, right? But in reality, what you have in a test tube, for example, is like you have many copies of your system, right? And so what we actually do is to only simulate a single copy, but we assume that it's surrounded by other copies. And there are ways to do this using periodic boundary conditions, which we're not going to talk about, but they allow you to simplify the process of representing what's happening in this test tube by studying a single copy of your system. So it makes it much simpler to run the calculations. Now, if you couldn't do this, that would mean you would have to then simulate each of these copies separately, right? Because indeed in a test tube, you don't have one protein, you have many proteins floating around, right? So okay, so you've actually seen this before in the last group kind of talked about this, right? So what's the scheme, the algorithm that's used to actually run simulations? Is there a question? Liz, is there a question in the chat?
Yes, we just got one. So Gal asked, so is the periodic box similar to the unit cell in crystallography?
I guess it would be an example of one kind of unit cell, right?
I
So
think it's more like an asymmetric unit. So it's more like the molecule you're interested in. So a unit cell is like everything that's within that periodic lattice,
right,
which is
it would be the,
constrained by the crystal context. So I think the constraints are very different.
but then the entire, because you can extend this infinitely, right, in all
So it
directions.
is an infinite lattice. So maybe it is a unit
Yeah,
cell.
yeah.
It's just the
So,
unit cell, it happens to be the asymmetric unit, which is rare.
yeah, but just to be clear, though, we only simulate one box. We just simply essentially use some mathematical tricks to take into consideration the possibility that this image, this central box is gonna have an image on the left, on the right, above and below, and in the front and in the back, right? And there are ways to impose those by keeping track of where the atoms are relative to the initial box. So what's the algorithm for running these simulations? It's pretty, in principle, very simple, right? Like if you were to imagine that you had a single particle that had was only able to move in one direction, let's say the x direction, you can actually do all these by hand pretty easily, right? So the first thing you need is a set of um, initial coordinates and velocities, right? And remember, the reason why you need this is because what you're essentially doing is solving Newton's equations of motion, right? Force is equal to mass times acceleration, right? And that is a differential equation, right? And to numerically solve differential equations, you need initial conditions. And so the initial coordinates and velocities are simply the initial conditions that are used to solve the differential equations. So you have some initial coordinates and initial velocities. Typically, you choose the initial velocities using some kind of a random selection process. The coordinates, when you're thinking about biomolecules, would come from X-ray crystallography or NMR. And then you initialize everything. You set your accelerations to 0. You set your time to 0. This is your counter. You set I to be equal to 0. And then you choose a very small delta t, right? And so what you do is then you you update your positions, right? Using an equation that looks like this here, right? So this equation should look very familiar to you. It's very similar to the equations you've seen in introductory physics, right? So your new position is equal to your old position plus your velocity times the time step that you take, right? Because you're assuming what's happening here is that you're asking after a time delta t, what's my new position, right? So to get the new position, you say, okay, I'm going to take my current position, add that to the product of my velocity times the change in time plus this half times the acceleration times the delta t squared, right? And that would give you a set of new positions. And then you can then update your velocities. So your new velocity is going to be old velocity plus the product of your acceleration plus your times your delta t, plus there are additional terms that we can neglect because we think they have small contributions. So now you have new positions and new velocities, right? And you could then go get your forces, right? The force is going to be equal to the negative gradient of your potential with respect to positions. And so that's why we need to have an equation that describes this VR, which is your force field. And once you have those forces, then you can get your acceleration for each of the atoms. So now we have new updated positions, updated velocities, and a set of accelerations that we can use. And you can apply this for the next step. So at this stages here, you can adjust your positions to account for any errors that might be associated with the approximations you might make. For example, in this part here, you have some additional terms that you neglected. You can update them here to kind of improve, fix those errors. This is the stage where you apply some boundary conditions. You can quantify your temperature and your pressure. And then if you want to calculate any kind of physical quantities, you can do that at this stage as well, right? Once you finish with all of that, you can then move forward in time, right? Because now you can set your time to be equal to t, what it was initially, which initially was 0. So now it's going to be t plus delta t. I is going to be equal to I plus 1. So initially, if I was initially 0, it will be now 1. And then you're going to go back to this step here and repeat it over and over and over again. So in the so in these movies that you've seen my primal, Liz, can you let me know if you've seen my primal? I don't have I can't see.
Yes, sorry, I was nodding.
That's fine.
Yes, we can see them, and there's a very lovely wiggly atom there,
Yeah.
or molecule.
So yeah, so in this case here, again, this is where we start. We have initial positions, so we know the location of each of these atoms here, right? So we have the initial coordinates. We randomly choose some velocities, and then we use them to update the positions, right? We have new positions, new velocities, then also new accelerations, update, keep doing this. And this is how we arrive at these kinds of trajectories. right? So that was the same question. So for those of you who might be interested in, right, if you have a little more details about how you integrate these equations, these equations, Newton's equations of motion, right? This equation here is simply, and essentially what you have is that your position with respect to time. So this is your velocity is equal to your momentum divided by m, right? Because you know momentum is equal to mass times velocity, right? So this is essentially what's shown in equation 4. Equation 5 describes the forces, right? The forces are just your negative derivative of your potential with respect to positions. And I should mention the RIs are your positions of a particular atom I. So using these two, you could rewrite the equation, Newton's equations as such. And then you could now come up with a numeric approximation that allows you to calculate your position, which you have now switched to as being x at some time t plus delta t based on the current position and this update part here, right? Which is kind of like we showed in this scheme here. So the point is that all of this kind of stems from Newton's equations of motion and applying a Taylor expansion to numerically approximate positions as a function of time. So what you can see is that the use of molecular dynamics has steadily increased, and so this is pretty old. This is an old graphic, but it captures the fact that in the early 1990s you had only less than 200 papers describing using molecular dynamics. In 2005, it's about 800, and now they're just this growth is probably exponential. It's not apparent from this curve, but I'm pretty sure it's exponential if you look at the current statistics. So how might we use it? So you could use molecular dynamics in principle to look at a number of interesting processes that someone who is interested in biophysics might be interested in, right? So one is transport of ions across a membrane. And so this is an example, the cartoon showing you a protein, and then you have some ion that's going to exit. And so you could use molecular dynamics to study the dynamics of this process, right? Because you could start simulations where you initially have the ion inside the membrane and look at exactly what events allow the ion to exit, right? So what kind of structural changes in the membrane protein allows this ion to be transported. You could also look at ligand binding. Here is a particularly interesting, important example because there are lots of drugs interact with receptors on the membrane, and so you can use simulations to study these processes. And based on what you observe, you can probably use it to guide drug development as an example, or just fundamentally understand, how does this receptor recognize this ligand? Now, conformational change, internal conformational change drives a lot of biological functions, so you can use MD simulations to look at those as well. And then just looking at protein folding, right? The protein folding problem can, in principle, be solved using molecular dynamic simulations. Can anyone guess as to what would be some of the challenges associated with using molecular dynamics to study protein folding, using it as a protein structure prediction tool? If you don't want to speak it up, you could just offer up a guess in the chat.
We have one in the chat now. Falling into local structural minimas instead of native structure. So getting stuck in some of the wells in the cone.
Yeah, so getting stuck in a intermediate, being kinetically trapped. But then why wouldn't you just wait for it to exit? Why does it even get trapped, right? So what does it mean to be trapped? What causes it to be trapped?
I have another comment. Without the library, too many sample changes makes it computationally expensive due to the entropy of the entire system.
Yeah. So yes. So there is a computational cost associated with simulating these processes, and so if it's a complex process or if you get stuck in a local minima, it'll take a really, really long time for it to exit, right? And so because of the computational cost associated with each time step, then it'll be really challenging to study protein folding in a reasonable amount of time. Now, if you had infinite amount of time, right? If you had the time scale of the universe, then yeah, you can use computer simulations. But most of us don't have that time, so it's not as useful. Yep. Any other? So a lot of what has been said so far is related to sampling, right? But a big assumption. What is a big assumption we impose here, right? Where do we get the forces from, right? Ultimately, the dynamics is dependent on the forces, right? And what's the input that gives us the force? Anyone wants to add that to the chat?
Could you repeat the question?
Oh yeah. So ultimately, the dynamics that we observe is dependent on the forces, right? The forces that we calculate. Where do we get the forces from?
We have one answer. The inputs come from the initial coordinates, which would make sense if which would only make sense if it's from some folded NMR or X-ray structure.
Right. Yeah. So that's one of the inputs, the initial coordinates. If you have bad initial coordinates, then it will take a very long time for you to arrive at a more higher probability state. But there's another thing that we need, right? So simulations require initial coordinates. Then we can also choose some initial velocities. But there's another thing we need. So remember, the forces are just a negative gradient of the potential with respect to positions, right? So if the positions are initially bad, then you can have a problem, but also if the potential is not accurate, right? So this energy function here then dictates the accuracy of the simulations, right? So if you don't have a good force field, then your simulations are crap, right? And the force field are really description of the functional form, right? So what do these equations look like, but also what the parameters are, right? So you have the parameters, for example, like KD and DO here. These are parameters that you have to determine before doing the simulations, and they will dictate the accuracy of the simulations. So
would some of those parameters deal with the environment in which you're doing this simulation, like in vacuum versus in solution?
So no. So those at times they can, right? But very often. So those parameters mostly describe the interactions between the atoms in assuming that there is no solvent. We add the solvent either explicitly by modeling water molecules in, or we might scale charges, for example, to account for the effect of water screening of charges. So yeah, so the reason why protein folding will be difficult using simulations is one, it's a computationally expensive process, right? And two, the force fields themselves are not perfect, right? So they can lead you to the wrong answer. So that means that the stable native conformation of the protein might actually have a high energy, right, or higher energy than a non-native structure, right? So these are some of the challenges that are faced, and it's just not the energy function problem is not only a problem for molecular dynamics, but also other protein structure prediction tools, right? So I kind of glossed over this a bit, right? But when I talked about this Monte Carlo simulations, I showed you a snippet of code from Rosetta. And here they're using a scoring function. You see this part here? They're setting up a scoring function. And the accuracy of that scoring function is going to dictate which moves you accept and which moves you reject, right? And so it will ultimately affect your ability to go from an extended state to the correct folded state. So even for structure prediction techniques in general, you do have the same kind of issue where if your energy function you're using that describes the interactions between the atoms in your system, if they have errors, then it's going to lead you to the wrong answer. So this slide here just kind of giving you a sense of a feel for the time scales that MD gives you access to, right? And that time scale relative to other experimental techniques, right? So all of them MD simulations can give you a time scale that ranges between femtoseconds to, I would say, microseconds, 10 to the -3. And that overlaps with techniques like NMR. NMR kind of has a pretty broad range in terms of time scales. And then you have FRET, which has a time scale between, I guess, microseconds to maybe milliseconds. So that's FRET. And so I guess the key here is the fact that MD simulations allows you to bridge a nice gap between some of the experimental techniques like NMR and those really fast processes. But also kind of highlights the fact that there is a limit, there's an upper limit on the MD simulation, right, a time scale. So if you look at some of the processes, biological processes that occur, right, light absorption is a really fast process, right? So that's not accessible by MD. You probably need to use quantum mechanics to do that. Electron transfer processes kind of overlap with the time scale of MD simulations, as does proton transfer. And solid permeation, right, is something that you can probably study using MD. Side chain flips, changes in secondary structure, active transfer processes. Once you get to active transfer processes, you're probably just about at the edge of the time scale that is accessible using MD. Channel gating, domain motions are still accessible. So anything beyond this millisecond time scale is inaccessible using MD. And so one of the big challenges or areas of active research is how do you extend the timescales of MD simulations? How do you go beyond, crack this barrier and go beyond the millisecond, right? So people have been doing a lot of work, right, in terms of trying to enhance the speed of simulations. And so here you're seeing a plot of the performance of molecular dynamics as a function of years, and it's given in nanoseconds per day. So what is it saying? How many nanoseconds of time can you simulate per day? The larger this number is, the better, right? And you can see between 2004 and let's say 2009, there was a pretty steady enhancement in our ability to simulate processes at a much larger time scale. And actually, the rate of increase actually outpaces Moore's law, which talks about the trend for the computing power to double each year. Now, here's one solution that has gotten a lot of buzz, right? And so there was this professor, I think he was a professor at Columbia in the computer science department, who left academia, and he went into he started a hedge fund, and he made tons and tons of money, right? And then after he got bored, he decided, well, I want to do something else. So he went back and he started building special purpose computers. So these are, what you're seeing here is the Anton chip as they're called, on the left, and on the right, you're seeing the Anton machine, right? So this is a computer that has one job only, to run MD simulations, nothing else, right? And so using their computers, they've been able to really extend the timescales of processes that they can look at, right? And so just showing you a couple of examples of processes they've looked at. For example, the transport of ions across a membrane. So they can, build these huge systems and all of the complexity of the system, simulate these dynamical processes, and has allowed them to really study the biophysics of this process. They've used it for protein folding studies, right? So you can see here examples of some relatively small, fast folding proteins that they've been able to fold. So they started simulations from the extended chain, and they were actually able to fold them to the correct structure. If any of you are interested in seeing some of these trajectories, you can just let me know, and I actually have some of their trajectories someplace, and I could share it with you. Interesting application of their simulation is using it to study ligand binding. So you have this drug here that binds, and so they actually started simulation where the ligand is somewhere in the extracellular space, and they are able to simulate this process, right? The entire process of binding, which is really, really cool. And you can see this is difficult, right? Because initially there are lots of places where this ligand can be, right? And so you can see in the extracellular space, there are lots of places it can be. And so it kind of samples a large variety of positions, and then ultimately it binds to the pocket, right? And it turns out that the binding mode, right? So if you look at this image here, in gray, I think is the actual position from X-ray crystallography, and in purple you can see the simulated ligand. And after about 3.68 microseconds, it actually docks into that location here. And so from these kind of simulations, you can look at measured distance or the RMSD of the ligand to monitor the process. And you can see initially it's sampling very large RMSD relative to its native position. So what I mean by that is that you can calculate essentially the distance between this ligand, the simulated ligand, and the actual experimentally determined position of the ligand. And the larger the distance, then the further away they are, right? And so you can kind of monitor the binding process by keeping track of this RMSD. And so you're seeing that here. So you can imagine using these kinds of studies to explore what happens when I make a change to the ligand, right? So if you made a change to this ligand, right, would it affect the time it takes to get to this location? Or would it affect the time it stays docked into this location? These are kinds of things you can do with these kinds of simulations. Now, if you want to do high throughput kinds of analyses where you have to look at thousands and thousands of ligands, then you probably wouldn't want to use these kinds of MD simulations, because even though they're faster, they're still computationally expensive. And there's only really, I think there are probably 2 or 3 of these machines, right? So it's not like we have tons of these machines everywhere. So for those of you, here are some summary points. You can take a look at this if when you're reviewing these slides. What I want to do next is to just give you a feel for how you experience in terms of running a simulation. It's very basic, but at least you would kind of get a feel for the steps involved. So while I get things set up, are there any other comments in the chat or questions in the chat?
So far, no, there are no more comments in the chat, but people can feel free to unmute themselves and ask a question or type it in the chat if they would prefer.
So I'm going to paste into the chat this website. Go ahead and click the URL, and I'm going to log out so that we see the same kind of screen. Give me 1 second. There's a log. Oh, I know. Let me use a private. Okay, so when you go to this URL, you should see the screen here. And so what I want you to do is to click on this NAMD button, and then you should see this page. So if you want, you can create an account, but I don't think it's necessary for the initial demo that we're going to do. But the first page just gives you some very basic instructions or information about MD simulations. And so what we're going to do is actually hover over the NAMD menu item and click submit a job. So let's just click submit a job. And then we're gonna click delete all molecules. And you should see something like this: no solute, just a nice refreshing box of pure water. We don't necessarily want that. What we want to do is let's click on add predefined. So we're going to add a predefined molecule. So click on this, and then you should see this this window, well, this sub menu pop up, and let's choose the molecule to simulate. So you can choose any one you want. I'm going to choose propane. And I guess you can choose also the number of molecules you want to use. I'm going to use 2 and see what happens. And we could do this in a water box, right? But it's going to take quite a while. So just to give you a feel for running the simulations, let's say no solvent, right? So we're not going to include any water molecules in the simulation. Now here's where we can choose what kinds of simulation we want to run. So it can be a constant volume simulation, right? So fixed number of particles and then constant volume, or it could be fixed number of particles and constant pressure. So you can choose any one you want at this stage. Then you need to choose a temperature, so which will define the kinetic energy. So it just says valid, need a valid. So okay.
Hey, Aaron.
Yep.
Did you see Jingxi's?
Yes. Yeah, I was gonna ask her to share her screen. Can you share your screen so I can walk you through this? And I'm pretty sure others might have a similar.
This sounds good.
Say it again.
This sounds good. I'm gonna do that right now.
Okay.
It just says that you have to use the PDB ID instead of the chemical name.
So what you do is, so can you scroll to the top again and try deleting in box number 1? Can you delete what you have there?
Yeah.
Delete the press delete on the right side. So
Okay. Right here.
Right. And then you go to predefined, right? So add predefined. So I could. There you go. Click on that. And then you can choose.
Oh, I see. Thank you.
Mm-hmm. Okay. So I said, let's not use solvent. So click on no solvent and you choose whichever kind of simulation you want to run, whether or not it's constant volume or constant pressure. And then you have to set a temperature. So this will dictate the kinetic energy in your system. So so one of you try setting the temperature to something very, very high. Maybe Liz, can you set it to something like yours to, I don't know, 500 or
I already typed 500, so I was on the same wavelength.
Okay, cool. And then let's do some minimization steps. So very often when you start a simulation, you want to minimize to remove any kind of steric clashes in your system. If you have any steric clashes, it can make the simulation unstable. So we have, let's mute someone. Yeah. So you do some minimization steps just to optimize and remove clashes. It's a pretty small system, so it wouldn't matter if I change this to a smaller value, but if one wanted to speed this up a bit, you can do that. Then you need to tell it how many steps to run, right? So how many times you want to update the positions. So in this case, it says 10,000 steps. And the time step is that delta t in the equation that we talked about, where you update the dynamics every delta t, and in this case it's in units of femtoseconds. So this here would be a case where we're using 2 femtosecond time steps. So once you have all that set up, you should be able to just click on submit, and you see some output in this window here. And because the system is really, really tiny, it's already done, right? Now, this is not typical for more complex systems where you include water, it's going to take a much longer time, right? But this one's done. So if you're working with a protein, you could actually visualize the trajectory in your window here. Let's see. But I can't seem to locate our molecule when I try this. So it is possible that it's just not rendering. So what you want to do is click on download output. So go ahead and download the output, and it will download a zip file to your local machine. And so we're going to assume you have PyMol installed in your local machine. And so I'm going to unzip mine. And what you want to do, I'm going to go start PyMol fresh. So open up PyMol. And in that zip folder, try to find the molecule you chose. So in my case, I chose propane, right? So I'm going to find that PDB and drag it over into my PyMol window. And then the object, the data structure that stores the trajectory is this md.dcd. So dcd is a file format. And so what you want to do is just drag this onto your molecule in PyMol window, drop it, and then it will ask you whether or not you want to load it, and we're going to go ahead and load that. So we have a problem here. So these are not the same molecules somehow. Let's see. I'll use another one that I had just so that I get rid of this. So I'll use the output from a previous run. So I think in that case, I must have ran simulation for pentane. I'm going to drag that here, find the MD .dcd trajectory, put it in here, and load it. Now what you would see is in some cases that the trajectory is not easily viewed in PyMol, is kind of shifted. So what you can do is type center in the PyMol window. And also you might want to type intra fit and then the name of the object you have here. So in my case, it's pentane. So I'm going to do pentane, right? And then center. So now I have my pentane molecule centered, and I can visualize the trajectory. So yeah, so are there any benefits? So this is really meant to be a teaching tool, just simply to give the uninitiated a feel for what it's like to what it's like to run a simulation and what the output looks like, right? So if you were wanting to do like production kind of simulations for research, this might not be the ideal tool. You can use it, but it might not be the ideal. But it's really meant to just give you a very gentle introduction to molecular dynamics. Another key benefit here is that you didn't have to install any kind of software on your local machine, right? So you're able to do this all in the cloud. So I want to try something. Let's see what happens if I run this with, oh, I already used two. That's what I want. Let's see if I run this and I use some explicit solvent. I'm not sure how long it's going to take, but let me change the number of dynamic steps to 1,000, and I'm going to change the minimization steps to 1,000 just to make it run faster. Liz, were you able to get your trajectory?
No, I had the same issue with you. I tried to put it onto my PyMol file, and it didn't actually do anything. It got an error.
Yeah, I think the reason for that was, let me actually just prove it now. I think because I set this to 2, I think that was the issue, right? So I said simulate 2 of these molecules, but the initial coordinate file that I loaded to PyMol just contained 1. So there was a mismatch. That's why I was saying there's 11 atoms in.
That makes sense.
Yeah. So I want to retry this just to prove that that was the issue. No. I guess I did this instead, but that's fine. And okay. So yeah, I think it was because I did 2 molecules. So what I want to do is actually try to see what happens if I do explicit solvent and take a much longer time, but let's try.
Yeah, I'm running an explicit solvent right now, and it's still running.
Yeah, so that's a good point, Pooja. And another advantage of using, another limitation of using these kinds of web interfaces is that you have limited functionality, right? So Charm and other MD simulation packages, they have tons of functionality, and it's very difficult to expose all of those functionalities in the context of a web browser. And so if you really want to do something fancy, like pull on some particular atom or do some kind of a complex calculation, then you really want to use the standalone version, or you want to actually use it where you can script it yourself. This here is just meant to give you a really, really easy interface to do very simple things, right?
Yeah, but there's something to be said, I think, about easy interfaces like this for introducing people into things like this and making it, especially the fact that this is free, is awesome for a science explicitness perspective.
Yeah, for sure.
I personally find computational and molecular dynamic simulations very confusing. I think they're fascinating, but they're definitely not intuitive to me. So if anyone has any questions, I suggest you ask them, because this is really cool and there's no stupid questions. But I guess if we just have 10 minutes left, do you want to talk about the extra credit project,
Oh
Aaron,
yeah,
while we're
yes.
waiting for
Thanks
these to
for,
run?
yeah, thanks for reminding me. Okay, so let's talk about the extra credit problem question. Okay, so let me do something. I need to grab a document. No, let's do this. So a while back, I was working on a project with a summer research student, and the project was centered around designing antimicrobial peptides. And so the details of how we design it is not as important, but I'll quickly just describe it. Essentially, it uses machine learning, and as soon as this page renders, I'll show you what that kind of looks like. But what we did is to design a set of what we think are antimicrobial peptides, right? And so one of the things we wanted to do at the end is to actually take those peptides and predict their structures, right? So the way the design works is that we have these machine learning models. We feed in some information in here about what examples of antimicrobial peptides, AMPs. And essentially what happens is that this model learns some latent space representation, which is just a fancy way of saying it learns how to compress the information in a sequence into a smaller data space, into a small space. And then you can do the opposite, which is to expand the information, and then that allows you to reconstruct your input, right? So what you can then do is then sample from this latent space, right, and generate new designs. And so that's what we did. So we generated a bunch of new designs. And there are these classifiers that you can use to classify whether or not the sequences are antimicrobial. So we had about 24 designs here, and we can use those classifiers to identify those that are predicted to be antimicrobial peptides. And so one of the things that we wanted to do at the very end was to predict the structure of each of these. And the reason we want to predict the structure is because the structure of these peptides actually dictate their function. So they have like a positive and a negative face, and that allows them to actually penetrate through the membrane of bacteria, and that's how they actually that's what gives them their antimicrobial properties. So given that we talked a little bit about AlphaFold, what I wanted to do is to give some of you the opportunity to kind of help us work on predicting some of these structures, right? So we have 24 designs plus two sequences for the native antimicrobial peptide plextocin that we're working with, and an analog. And so for each of them we want to predict their structures, right? So if we have a subset of you who are interested in this project, we would set up a Google Doc where you can choose which one of these peptides you want to predict their structure, or we'll probably assign you those. And then once you get this structure using AlphaFold, we can figure out whether or not any of them fold into any unique conformation. And if they do, these would just be the structures or the designs that we want to go ahead and test in the lab. So for those of you who are interested, what would be the easiest way to do this? I'll send out an announcement either today or tomorrow describing the logistics. So I'll send out the exact sequences and figure out how to assign you to a particular design. So what else? So for now we can use the AlphaFold, the Colab notebook to do the predictions. If that becomes a problem where it's too slow, there is a possibility that I might be able to implement or design a web interface that connects to one of our computers that you can actually use. But let's first try using the Colab notebook, and this will serve as the extra credit project for the course for those of you who are interested in. Any questions?
My simulation just finished.
Adjustments?
Yes. So I'm gonna open my PyMol real quick.
Okay, cool. You go ahead and share screen. Did it finish successfully? So that's the thing that we always get, right? So it's one thing for a job to finish. There's another thing for it to finish successfully. So, without error.
Okay. Oh, my trajectory file didn't load again. Oh, it's because it has waters in it. Do I have to add water? How do I do that?
Sorry, if you share screen, I might be able to show you.
Actually,
So what you did is that you tried to load you tried to load the
just
trajectory
let
of
me share
water.
my screen really quick. Yes, so I just loaded the file. Let me share my, I apologize, my screen is full of windows. So I pardon everyone who likes a really clean desktop. Okay, so here's my PyMol window. So what I did is I went down to my PDB.
Yeah, you can't use that one because it only
I
contains.
can't use that one.
Yeah. So it's very tiny, but let me see if I could.
Oh, I can make it bigger.
So I think it's still. So you see the md.core file.
This one.
It might be a PDB file that's masquerading as a different kind of file. So if you change the extension to .pdb, you might be able to load it into PyMol.
How did you delete using the command? It's like asterisk delete.
Or delete just delete then the asterisk. Yeah.
Okay, we'll see. Oh my gosh, there's so many waters.
So now you could load the md.dcd into that. It's not giving you an option to load.
Oh no, it just did. But
yeah. So go ahead and click play. And yep, that's it.
Awesome. Sweet, that's cool.
So yeah, it takes a little longer to run, and for such a small molecule to be surrounded by so much water molecules is a kind of wasted computational effort. But that's kind of just to give you a feel for how you would actually run one of these simulations and what the output would look like.
Neat though.
Yep. Okay. Any more questions, or if you have any questions, just shoot us an email and we'll answer those. But I hope you have now a better feel for what MD simulations are and how they might be useful. So see you guys next week, Tuesday.
Oh, Aaron, will we be in person next week?
Yeah, next week we'll be in person. I should have said it. So next week, Tuesday, we'll be in person. So
okay,
we lost.
I can send an announcement about that.
Yeah, thanks.
Yeah, of course.
Was someone saying something? Oh, no,
I think someone said thank you.
Okay, cool. Okay.
Cool.
See you next week. Well,
I'll see you tomorrow. But
tomorrow, yeah. Are we doing it in person or meeting in Zoom?
I think we're gonna meet in person. I was gonna reserve a conference room, but we can meet in Zoom if that's better because we might want to be sharing stuff. So
yeah, maybe because then I have I have a bunch of meetings tomorrow too. So
do you have mostly Zoom meetings tomorrow?
Yeah.
Yeah, then maybe it'll be easier for us to do it on Zoom.
Yeah, find out what Marcos prefers.
Well, Marcos always prefers in person. So I think that you should say what you prefer and don't be bullied by Marcos.
Bullied. That's funny. I'm gonna have to delete
Well,
this.
he's not actually bullying you. He's
I'm
just
gonna have
got
to
strong.